Okay, welcome back, everyone. To start the next section, I'm excited to introduce Professor Joe Handelsman, who is the director of the Wisconsin Institute for Discovery, which is one of the two institutes right here in this building. So after diving deep into many different aspects of mass spectrometry, I think for this next session we'll be zooming out a bit more. We wanted to have some breadth in the program and add some lectures that were provided some perspective beyond the details and the latest and greatest in mass spectrometry. So Joe has a long history here at UW-Madison. She did her doctoral work here and was both a faculty member and a department chair for many years before leaving for Yale in 2009, where she became a Howard Hughes Institute professor. And also in the 2 years before coming back here, Joe served as the associate director for the U.S. Office of Science and Technology Policy under the Obama administration. So in addition to Joe's scientific excellence, she's well known for her work in science teaching and mentoring, and also in her work in understanding implicit biases that affect scientists' attitudes and behaviors. So today she'll be telling us about some exciting science work, not more on the policy side, but related to antibiotic discovery. So, Joe, welcome.
Thanks. Oh, okay. Well, thanks, Dave, and thanks everybody for being here. This is going to give you a little bit of variety, as Dave said, since I'm going to talk about the fusion of education and science, and a somewhat new initiative, not brand new, but a few years old, that we're hoping will really change the landscape on small molecule discovery. So the initiative I'm going to talk about is called TinyEarth, and it's based here at the Discovery Building upstairs. It's an educational initiative that also involves research, and it also addresses dual challenges. So its nature seems to be dualities, education and research, and then dealing with a world crisis on the lack of antibiotics to treat infectious disease, as well as a world crisis on generating sufficient STEM workforce members. So I want to just talk about those two crises for a minute. This is an example of the kind of untreatable disease that is developing in this country. This is US data on MRSA, which is methicillin-resistant Staph aureus, an insidious pathogen that has become more so because of its multiple drug resistances. And so this is cases over about an almost 30-year period. And you can see the increase in frequency of these resistant cases is going up to, it's almost 400,000 cases per year in the US today. Worldwide, the WHO and CDC have predicted that the leading cause of death from disease will be infectious disease that results from antibiotic-resistant bacteria by the year 2050. And so some of their projections are that this is not cases, this is deaths. So now we have, for example, with MRSA, we have about 400,000 cases per year. They're predicting we'll be up to over 300,000 deaths per year. And you can see across the world, we're talking many millions of people dying. Today, I think the antibiotic-resistant bacteria are down around the third or fourth leading cause of death, but this is rising all the time because we're losing effective antibiotics from treatment because of resistance developing. Simultaneous with that is the lack of drugs in the pipeline. So in the 1980s, when antibiotic discovery and release of commercial antibiotics was in its heyday still, from about the 1940s during World War II until the 1980s. There was an enormous drug industry dedicated to discovering new antibiotics and commercializing them. And you can see the number of new antimicrobial agents that appeared on the market. But since then, that pipeline has dwindled, and in the last period that we have data for, it had dwindled to one antibiotic being released on the market in a 4-year period. And the last 4 years, I believe, we're at 1 or 2. So this is two conflicting trends of more resistance among the pathogens, so they're not responding to the antibiotics we have, and fewer antibiotics being discovered in industry and released on the market. It's sort of disturbing that it seems like every couple of years we lose another member of the major pharma companies from the antibiotic discovery area. And for a long time, there were about 6 big companies hovering in this area. And then there were 4, and when I went to Yale, I was very excited because Pfizer was right down the street from where I was going to be working. And the month after I got there, Pfizer pulled out of antibiotic discovery, and then last month, Novartis pulled out. So we're now down to 2 major pharmaceutical companies still working on antibiotic discovery. Fortunately, there are some young startups, but right now the pipeline from those has not been established. So we are facing a very serious crisis, and we're not doing very much about it. The government tried to develop incentives for pharmaceutical companies to get involved in antibiotic discovery, and that just hasn't worked. Those incentives have been in place for about 12 years, and we haven't seen very much change in the amount of R&D in industry. One of the problems with antibiotics is they're too effective, is that you take them for 5 to 10 days and then you're done. And so there's a lot less profit to be made for the same amount of R&D and regulatory work as a drug like a cholesterol drug or a heart disease drug that one takes for the rest of one's days. Those are much more lucrative drugs. So we can't really blame the pharmaceutical industry for moving to the more lucrative drugs. That is their job. But it leaves us in a real predicament as a humankind. We're threatened with going back to the pre-antibiotic era, so through the 1920s and 30s when antibiotics were rarely, if ever, used. And that was an era where most of the top killers were bacterial diseases. But it's going to be much worse than that because our populations across the world are so much more dense and so highly mobile. They move across the world that epidemics will spread much more rapidly in local environments and then across the world. So this crisis is one of the driving forces behind TinyEarth. The second one is similarly urgent because it affects all areas of science, technology, engineering, and math. And that is that we're facing a deficit of STEM workers. And the problem here has been obscured by what's seen as an excess of PhDs. Here we're talking about the bachelor's level trainees. And what was found in a government report that was presented to President Obama in 2012 was that from 2012 to 2022, we expected a rise of about 1 million jobs that wouldn't be able to be filled in this country due to the lack of STEM-trained workers. We're now at about 500,000, so just as those projections indicated, that population is becoming scarcer and scarcer. So this is a great time to be getting into STEM fields, but at the same time, our students tend to leave STEM fields at incredible rates. And one of the kind of scary statistics is that of all the students who start in a STEM field, they start college in a STEM field, intending to major in any one of the science and technology fields, 60% of them will switch into a non-science field, either social science or the humanities, during their college years. And so we're not capturing the talent that's being offered to us at the college level. One of the reasons that this is such a concern is that the grade points and the performance in their STEM classes of the students who leave are similar or identical to the students who stay. So it's not like, we often say in science, we have these weed out courses that we're weeding out the ones that don't belong there. In fact, we're weeding out students who perhaps would be outstanding scientists and engineers, but we bore them to tears in our introductory courses. And all of the studies that have looked at that shows that that's why students leave. And if you look at the performance of students who have gone on in other fields who had been doing well in STEM majors, many of them go to graduate school and have very successful careers in their other fields. And one of the very common comments is, when asked, why did you leave science or technology fields, they say, because science is about the what, and I'm interested in the why. Now, how ironic is that, given what we all do as scientists? Of course we study the why. So there's a problem, there's a disconnect between the way we teach science in our introductory courses, which is all about the what and has students memorizing lots of information and learning a lot of concepts about what has already been done, but doesn't really provoke them to think about the why sufficiently, or about what could be done in the future in terms of research. And so TinyEarth came about when I was a professor at Yale, and I fulfilled a teaching dream, which was to teach introductory biology through research, with the idea that you would take freshmen and give them a freshman biology course. But the reason they would be learning all the facts and concepts would be because they were doing research, and they would need that information in order to do the research. So in other words, they'd be learning the way all of us learn. We learn because we go to the library or the internet because we're doing research and we have a question and we need an answer and we need to understand some new concept. And that is one of the most powerful driving forces for learning new information. And so the idea was that we would give freshmen the excitement of discovery that would not only get them excited about being scientists, being researchers, but it would also give them the wherewithal to survive those boring introductory courses. So I thought, well, if we can't change the introductory courses, maybe we can make our students more robust, more resilient, more able to deal with those courses. So it's a discovery-based introductory microbiology course. It can be used and has been used as a general biology course for majors, for non-majors, as a standalone research course. It has been shown to inspire students, and research courses generally have been shown to have a tremendous impact on retention in the sciences, and also retention in school and an increase in grades. So research courses early in college have a tremendous impact on our students. And one of the things that students find very inspiring is that it addresses a real world problem, this antibiotic resistant bacteria challenge that we're facing today. And it also sets up a crowdsourcing model for research and discovery that I think has a lot of potential for other kinds of research, other areas, and other discovery. So the process is pretty simple. The students go out and collect soil from a place of their own interest. Most of the antibiotics we have today, about 75% of the antibiotics that we use in clinical medicine, come from soil bacteria. So we're going to the place where we know antibiotics are very richly distributed. They culture bacteria from soil, so they end up with plates, culture plates of diverse organisms. They test the bacteria for antibiotic activity. This is an example of a zone of inhibition. That's the assay that they're using. They then take the producing organism and they sequence the 16S ribosomal RNA gene, which gives them a phylogenetic and taxonomic tool to tell what kind of species they're working with. And we do this for two reasons. One, it's the most commonly used tool for taxonomy today. It works on essentially the entire biological world. But it also tells us very early in the course whether they're working with a dangerous organism. And so if they are, then they don't continue with that particular isolate if it turns out to be a pathogen. We have right now about 10,000 students per year taking, TinyEarth, taking the TinyEarth course worldwide. And the workflow has been tested over and over in hundreds of classrooms and seems to work routinely and very reliably. And essentially in every course that I've seen, essentially every student will get antibiotic-producing bacteria. So the question then that I'll talk about in a few minutes is how do we go from activity to chemical structures and figure out which of these are novel compounds. We offer an instructor training, and that has been an important element of TinyEarth because we've trained all of the about 280 instructors across the world who are teaching the course, and that means that they all have the same experience. We have some quality control over what's taught. We give them books to teach with, but we make sure that they've been with us for a week with a workshop where they do all of the experiments that the students will do. And we can then ensure that the students are working with a common set of methods and a common set of standards. It also provides the instructors with a network of other instructors, and that also approximates science, just like the course itself does, that all of us, as this week has, of course, been a great example of, use each other in the scientific community to troubleshoot, to brainstorm, and to improve our science. And the instructors have that opportunity. And even though teaching should be more of a social occupation, it really isn't. Most of us teach in pretty much isolation. We don't really talk to our colleagues very much about the content or practice of our teaching. And the instructors tell us frequently how important this network is to share with an instructor thousands of miles away the same teaching experience and solve problems together. So the growth of TinyEarth has been pretty exponential over the last 6 years. We're now up to 280 schools and 14 countries outside the US. We're in 41 states across the US and in just about every kind of institution you can imagine. So we have partners at places like UW, MIT, Berkeley, and then we have 4-year liberal arts colleges. We have 2-year campuses like we have in Wisconsin. We have 2-year and 4-year campuses. 2-year colleges are often called community colleges or junior colleges in other areas, and then also in tech colleges. And we've even included some high schools. This year, just last week, we had a training for high school teachers from Milwaukee and New York City, from a high school on Staten Island. And so far, the implementation in the high schools has been very effective. They often will use a slightly slimmed down version of the course, but if they come to the training, then the instructors usually find it's pretty successful. So our distribution is pretty wide geographically, as well as the kind of institutions, public and private, rich and poor, that students can take the course in. This is just an example of the mix of students and how well it works. These are students from a combination of our local area technical college and students from UW-Madison. And they got together at the end of last semester. They were, both groups were taking the course, and they compared notes. And it had a lot of social implications for them. One of them being for the students at the technical college who wanted to transfer to Madison, it gave them enormous confidence because here they were having conversations with UW-Madison students and realizing they were doing the same curriculum, and they were able to have fully open and intelligent conversations with these other students. So it has a very normalizing effect. But at the same time, there's nothing watered down about it. This is real research. So we think that there's a real resource here. With 10,000 students a year taking the course, and that's rising all the time, we're hoping to reach 100,000 students in the next few years. We have this resource of soil samples and microbes coming from those samples, and microbes that produce antibiotics coming from them, from all over the world. And so that's quite a resource to use for antibiotic discovery. So a lot of people who know the antibiotic industry are familiar with the arguments that we've heard since the 1980s, when the antibiotic industry kind of departed from antibiotic discovery. They have argued that the soil is tapped out, that we're all done. And so we've had to think very hard about the rationale for the course, because we're going to obviously the most tapped environment, because it's always been the richest source of antibiotic-producing organisms. My evidence would suggest, and I think there are others who have looked at this and would agree, that the soil is in fact not fully mined. We know that we only know about 1% of the diversity of organisms in soil, and a lot of those are not culturable, so they won't come up on the plates that our students work with, but many of them are, and we're discovering new organisms all the time. But we also think that there are reasons to believe that there will be many new natural products. And so these are just two examples from my lab, and these were both found when we weren't looking for antibiotics. We were looking at some other problem, and these antibiotics turned out to be the answer to them. So this is one produced by Pseudomonas coriensis. It's a group, a family of compounds called the coriansins, and they're alkaloids that resemble some of the alkaloids from plants like hemlock, the conicines and conine type compounds. And these are a really interesting class of antibiotics, because they only affect one particular phylum of bacteria, and it's a phylum that there have not been many antibiotics directed toward. Early on, my group discovered Bacillus cereus, one of the most common soil organisms. There are about 10 to the 5th Bacillus cereus per gram of soil in just about every soil we've ever looked at. So these are extremely common organisms. And about 10 to 20% of them produce this novel antibiotic called Zwittermycin A. And it turned out to be a new class, an aminopolyol, new class of antibiotics with a very unusual set of characteristics. One, it has a novel biosynthetic pathway that was the basis for discovering 2 new starter units for polyketides. So anyone who knows polyketide chemistry, these are compounds that have these large biosynthetic complexes that usually start with 1 of 4 starter units, and there are 2 new ones in the Zwittermycin pathway. So this is a pretty exotic organism and pathway that was sitting under our noses all over the world. Interestingly, also, the activity range of this antibiotic is extremely interesting, because it affects both bacteria, gram-positive and gram-negative bacteria, so the 2 main divisions of bacteria, and then oomycetes, which are protists that infect plants and cause disease. So that's where it's been studied the most, because it has some very interesting properties for controlling plant disease. So this is just a couple of examples. My lab's discovered others, but these were just 2 antibiotics or classes of antibiotics that we stumbled upon. And so these would indicate that the soil is not tapped out. Bacillus interests me the most because it was so abundant. Every soil we looked at had plenty of bacillus, and we found there were, in most soils, about 10 to the 4th per gram Zwittermycin-producing bacilli. And it's one of the most readily culturable organisms in the soil. So the question is, why didn't the antibiotic industry ever discover this? And there are a few reasons we think that might be true, that might have caused that, but we can go into that later if people are interested. So if we basically accept the premise that the soil is not completely mined, then what else does this network offer us? One of the things that, and this would be true even if the soil had been fully mined, is that most of the mining of antibiotics from soil has gone on in private industry. And therefore, we don't have access to the probably 50 or 60 years of very intensive searches and the data from those searches. And so we don't have a lot of information about the trends and the frequencies of where you find antibiotics, what kinds of soils, what kinds of conditions for isolation, because a lot of that was done in industry and never published. And so even if we simply repeated all of that, what has been done in industry and published it, we think that this would be an enormous contribution and a database that really will be useful for asking many, many questions. But we also think that we're working with a tremendously diverse set of soils from across the world. The methods that we use are not necessarily those that the antibiotic industry used. And we have our wild card factor, and that's student ingenuity. And we know from copious research that diversity of people leads to better thinking, better solutions to problems, and a more diverse set of solutions to problems. So we think that this sort of crowdsourcing could lead to a creativity, a level of innovation that we don't see when we work in a single laboratory or under the normal paradigm of science. And so here's just one example of what we found using student discovery so far. In my first class, I only had 6 students take the course the first time I taught it at Yale, so I thought it was going to be a complete failure because only 6 students took it. But the 6 students who took it really loved it. And now, of course, there are 10,000 students a year taking it, so it hasn't been such a failure. But one of the successes that came out of that was one of my Yale students noticed that when she plated her soil on a medium called PDA, potato dextrose agar, she got a lot more antibiotic-producing bacteria than when she plated on 2 much more common media, tryptic soy agar and laureate broth. So that was kind of an interesting discovery, and it was striking because in her hands it was about twice as often. She'd find twice as many antibiotic producers on PDA than the other media. But this medium came out of my history of working for many years in a plant pathology department where PDA is a common medium used. It's used to culture several of the fungal plant pathogens, but it's not something that most microbiologists have ever heard of. When I moved from plant pathology to bacteriology, my colleagues said, "What's that?" So this is not something that the typical microbiologist in industry would have used for the 60 or 70 years of antibiotic discovery that went on. So we recently went back to this student's result and quantified it. And we found, so for example, here we're looking at inhibition of Pseudomonas putida, which is just one of the benign organisms we use as a test organism. And you can see the same isolates on 3 different media with Pseudomonas putida, and it's pretty noticeable that on PDA, you can see the zones of inhibition are much more striking than on the other media. And so we quantified this in just one experiment. We found, once again, that PDA gave us consistently more antibiotic producers. And once again, it was about between 50% and 100% more antibiotic producers. So we're interested in why this is. Is it that we're isolating a different group of organisms, or that they don't produce antibiotics on PDA or on the non-PDA media, and they produce on PDA? And it seems like it's a combination of the two. When we did a 16S taxonomic analysis of the members of the community we isolated on PDA versus TSA, they were almost non-overlapping. So very, very different species were coming up on the 2 media. But this is an example of an organism that would grow on both, but it does seem to be differentially expressing its antibiotic pathways. on the media. And so this is Erwinia endophytica. And this is one of our most interesting candidates because it's not a species that was ever reported to produce antibiotics. So even though it's readily culturable from many soils, it has not been isolated as an antibiotic producer. So we think that that's intriguing in itself because it probably has been cultured many thousands of times, and if no one ever found an antibiotic in it before, perhaps we have something new. We also have looked at the total genome sequence, and we find no sequence similarity to known antibiotic biosynthetic pathways. And the antibiotic is produced only on PDA and again not on the more standard media, suggesting that it could very easily have been simply missed when the antibiotic industry was screening. So we think that using the diverse soils and unusual methods and then student variations on the theme will lead to more discovery. But even if none of these none of these other elements contribute to antibiotic discovery science, our numbers are exceptional. Since we are already at 10,000 students, and most students will come up with about 10 antibiotic-producing isolates, we can pretty readily, as a network, isolate 100,000 antibiotic-producing bacteria per year. So these numbers will very rapidly match or exceed what was done over many decades in industry. And the question then is, how do we decide what to study from there, and which ones actually contain new antibiotics and which ones contain old ones? So to understand that and get an answer, we are creating what we're calling the Chemistry Hub here in the Wisconsin Institute for Discovery, right upstairs. But it's going to be a distributed network with its headquarters here. Our three starting partners are Jason Crawford, Marcy Balunis, and Peter Dorstein at Yale, UConn, and UCSD, respectively. They're all chemists who are very adept at very rapidly dereplicating compounds, figuring out which ones are old compounds, known compounds, and which ones deserve more attention, and then determining structures of the new compounds. We're hoping, according to Dorstein, he thinks this is completely reasonable, that we'll be able to crank through between 10,000 and 40,000 extracts from the isolates from around the world per year. So the question is how to prioritize. So what do we look at? Because the chemistry is obviously the hard part of this whole discovery process, and particularly the antibiotic purification process. So how do we decide which ones to study further and which ones are worthy of purification? So the first criterion is that we're working with a species or a genus that doesn't have any known antibiotics in the literature. So that is a small indication, not a guarantee, that there might be something novel there. We look at the solubility and activity profiles, and some of the solubilities are unusual. That was the case with zwittermycin. It's extremely water-soluble, probably more polar than most of the chemists in the pharmaceutical industry wanted to deal with. In fact, we had a hard time finding a chemist to collaborate with because nobody wanted to work with a water-soluble molecule. So that's an example of where it may be harder, but we also may find more unusual structures. And then looking for interesting activity profiles. One of the things about the history of antibiotic discovery is that traditionally companies wanted to go for broad spectrum antibiotics, antibiotics that would inhibit many, many different groups of bacteria. We now know that that leads to much more rapid development of resistance because the antibiotic is used in many more situations. So the trend now is to find much more highly specific, narrow spectrum antibiotics. And we think that by looking for those, we will also be increasing the chance of novelty because those are exactly the extracts that the pharmaceutical industry would have deprioritized. So based on genome sequence, we can get a lot of information, and the first criterion is looking for no known pathways, and we have other criteria as well. This is going to be a powerful tool because we have a collaboration with the Joint Genome Institute, which is a Department of Energy lab in California that is doing high throughput sequencing for us, total genomes of what we hope will get to be a few thousand of our isolates per year. And so that gives us incredible insight. In addition to the activity and chemical profiles, we also can use genomic sequence information. And then finally, the step that I think is the biggest revolution in my experience of natural products chemistry in the last probably half century is the use of mass spec profiling on complex mixtures. And so this has been pioneered by several people, but in particular the Global Natural Products Social Molecular Networking project at UCSD that's headed up by Peter Dorstein. And there we can use profiles of complex mixtures and then use a computational analysis that Peter's group has developed to identify those mixtures that have potential for discovery. And so he has a great program that will pull out mass spec features that look like they're unusual or ones that match known compounds and probably don't merit a lot more attention. So that's where the speed and rapidity of our analysis can come in. So this is the TinyEarth headquarters team. So Sam Rickers is the executive director of TinyEarth at Madison. Nicole Broderick leads the training of all the new instructors. She's at UConn. And Simon Hernandez is now in medical school, but he was the undergraduate and staff member who helped me write the books for the TinyEarth that are now used for all of these students and their instructor training. So with that, I'd like to stop and take questions and invite anybody who'd like to be a partner in TinyEarth to join our search. Thanks. Yeah, those are two great questions. So the first is about getting the information public. So we have had a database that was very, very simple that all the students from around the world would put their data into. And we're working with a company nearby that is developing a much more sophisticated database that will be much easier for the students to use, so we're hoping more of the students will actually put the data in, but also will be much easier to use machine learning and other techniques to analyze. And so we're actually developing a course here at UW on machine learning for undergraduates that's going to use the database as the basis, and we'll see what we find. So that will be the primary source of the raw data. In terms of publication of more finished products, there have already been a few publications, so individual instructors will publish with their students. We're also developing a publication from here that will engage several of the different nodes of the network and several instructors and students. So that seems to be kind of an organic process of publication. With the Chemistry Hub, it may get more complicated or it may be more routine. We're not sure yet. What we're planning is that when new compounds are discovered, the staff of the Chemistry Hub who determine the structures will collaborate with the instructors who led the students who originally discovered the organisms and have a joint publication with those students, instructors, and the staff of the Chemistry Hub. Intellectual property, which is part of your second question, and then ultimately commercial development. We've settled by saying that any structures that have been determined or compounds that ultimately are patented will reside with the Chemistry Hub, And so the chemistry hub is here, so it will be WARF, which is the patenting and licensing arm of the University of Wisconsin. And the agreement is that if there are ever royalties or license fees from products, then those will go back into the TinyEarth network. And that will be described very broadly so that we can set up more chemistry hubs, enrich classrooms across the world, bring more students excuse me, to our annual symposium. Use it for all sorts of purposes so that, because of course WARF thinks in very positive terms and said, well, what if you have $1 billion drug? What are you going to do with it? So we're going to make sure that the language is broad enough that we can enrich research worldwide. So that's the plan on IP. And we've already started having many institutions say they're going to sign our material transfer agreement and send their isolates to us. We're working with a couple of large pharmaceutical companies, a couple that are still remaining in the business, on potential funding and then ultimately commercial development of antibiotics. And what we've heard from other companies, not the ones that currently are in the antibiotic business, is that they would be interested if they didn't have to do the groundwork. So if there were novel chemicals and the activity spectrum was known and the properties of the molecules were all known, then they would get interested. So we think that this is a pretty inexpensive way to do their R&D for them and take the molecules to the point at which they will engage. And so we think that there's a lot of opportunity there for commercial development. And if not, we'll form a nonprofit company or something and figure it out ourselves.
Hi, thank you so much for that presentation. This is so inspiring and so cool. I don't teach introductory biology or molecular, what is it? The thing that, with the critters. I teach introductory chemistry. And I also see the sort of glazing over and the attrition, and it concerns me as an instructor of introductory chemistry. I'm wondering if you've had any thoughts or any conversations about how a similar sort of model, perhaps even partnering with TinyEarth, could be brought into introductory chemistry classes to bring this active research pedagogy into chemistry also.
Yeah, great question. I was hoping a room full of chemists would ask that. So when I was at Yale, Andy and Jill Phillips, who are two chemists who were then at Yale, now they're in Cambridge, said, oh, we could develop a chemistry course that would be the follow-on. You teach the biology in the fall, and then the chemists will pick it up, and we'll teach them organic chemistry in the spring. And we found that it just didn't work for a variety of reasons, but their argument was that by working with new molecules, you couldn't tell what pace, at what stage the students would advance to during the course. And that severely limited the number of different methods that they could learn because you couldn't predict how long one step would take or another. And so we were kind of discouraged. They didn't want to teach it again. They said in the end, they actually gave all the students a known antibiotic and had them learn their spectroscopic methods on that. And that, of course, defeats the whole purpose. So that just didn't seem successful. So I was actually kind of negative about chemistry courses ever being able to do this until I talked to a chemist at Oklahoma a few months ago. And she said, with the advent of the computational side, you have the opportunity to, even if the students never get a structure, if they just learn something about chemical extracts and mass spec and computational analysis, they've learned a lot. And so if we start setting our sights there, I think a course becomes extremely manageable and very rewarding for the students. And so we're working with the Howard Hughes Medical Institute to get funding now for that course, and we're going to be developing it. So if you want to be a partner on developing that course, do let me know, because we definitely want partners to give us ideas.
We'll take one more.
Your network in hand. Have you ever considered
Yeah, so the arms race is obviously the concern. We're always going to be dealing with resistance. That's just a product of Darwinian evolution, no question about it. We hope that by having more antibiotics and perhaps more narrow spectrum antibiotics, we will reduce the rate of evolution of resistance. But we're not expert as a field on how to do that yet. So I think that's still a work in progress. Bacteriophage have been isolated for controlling infectious disease for quite a long time in Russia and Georgia, in the former Soviet Union. They were developed about 100 years ago and are still used today for controlling infectious disease. For a long time, they were not popular as a tool for treating people in this country for reasons I've never understood, except that perhaps the antibiotic industry was so successful that we didn't need them. Now people have gone back to them, and I saw some data recently from an NYU microbiologist who has clinical trials with a phage-based enzyme that digests the wall of bacteria. And it was some of the best data I've ever seen in any field, but it was almost 100% cure rate of bacterial infections. So I think phage are starting to come back into vogue in this country. There is a course that actually our TinyEarth was based on, which was phage discovery, but it didn't have any obvious medical implications because phage weren't, at the time it was developed, really being used in a medical arena. And so that's why we developed this, actually, was because we thought it was more exciting for students to work on something that had more immediate implications for human health, and also just because I like chemistry better than I like phage. But yes, phage are definitely an alternative, and I think you're going to see more and more reports of them.
Fantastic. Thank you so much.